We have already met DNA, the blueprint that makes us us. It contains the information for how to make and run every living being. And this information is present as a sequence of nucleotides. But DNA, being super precious, is locked away in the nucleus. That's where mRNA or the messenger RNA comes in. mRNA is the working copy of a gene. And the mRNA, being the working copy, can leave the nucleus anytime it wants, carrying along instructions for how to build a protein. Now, like DNA, mRNA also carries information using nucleotides. And it works with 4 nucleotides: adenine, uracil, cytosine, and guanine. Proteins, on the other hand, are made of totally different molecules called amino acids. So they are speaking a whole different language than mRNA. So how do we go from one to the other? Well, that's where translation comes in. And mRNA translation isn't exactly like going from English to Hindi. So come, let's get a better look at how it works. Now for simplicity, consider this is our mRNA. And the nucleotides you see here are written in groups of 3. Do you know why? It is because the nucleotide alphabets on mRNA are always read in groups of 3. These 3-letter words are called codons. And with a few exceptions, each one represents a possible amino acid. So the letters G, U, A together represent an amino acid called valine. UCC, on the other hand, stands for serine, so on and so forth. There are 64 possible combinations, which makes for a manageable genetic dictionary. By the way, those exceptions I mentioned, they are called the stop codons. They are like full stops, telling us when a genetic sequence ends. Now the cellular machine that makes sense of these nucleotides as codons and translates them into amino acids is called a ribosome. It puts together amino acids to build a peptide. Ribosomes themselves are made of protein and a special RNA called the ribosomal RNA. Now, if you look carefully, do you see how the ribosome is in 2 pieces? Those 2 pieces are the 2 subunits. One is a large subunit and the other is a small subunit. When ribosomes aren't actively translating, they spend a lot of their time separated like that. But translation involves the 2 subunits working together. First, the small subunit of the ribosome binds to the beginning of the mRNA strand. The start of practically every mRNA has the same 3 letters, AUG. It's the start codon. And it codes for the amino acid methionine. Methionine is a key ingredient that's needed to start building a peptide. But the ribosome doesn't just go off and fetch the methionine itself. That is where the transferred RNA or the tRNA comes in. You can think of tRNAs like delivery truck of the translation world. There are dozens of types, and each is specialized to carry and install a specific kind of amino acid. Now, one end of the tRNA carries the amino acid itself, and the other end has 3 unpaired nucleotides. They are called as anticodon. And this anticodon is complementary to the codons on mRNA. Now for simplicity again, let's replace this doodle of an actual tRNA with something like this. Great. Now tell me, what would an anticodon on a tRNA carrying methionine look like? The codon for methionine is AUG. So the anticodon on tRNA would be UAC, right? And once the tRNA arrives with methionine, it clicks into a place on the mRNA, letting the complementary nucleotides pair together, like 2 ends of a matching puzzle piece. And that's when the large subunit of the ribosome clicks in, ready to start building proteins. Now, as new tRNAs come in, the ribosome moves along the mRNA 1 codon at a time. With every new codon, a new tRNA brings in the next amino acid. And the ribosome links it to the growing chain with what we call as a peptide bond. It's like clicking Lego blocks together, joining amino acids in the right order. Eventually, the ribosome hits a stop codon. No tRNA matches these. So the ribosome knows this chain is done. It lets go of the peptide chain and splits back into its two subunits. Translation is complete. But wait, we have still not made a protein. We have just made a peptide, right? A short chain of amino acids. To become a full-fledged protein, it needs to merge with other peptides. Only then can it go off and do big things like build muscle, digest food, or fight off germs. And with that, we end the story of translation. Let's do a quick recap. mRNA is read in codons, and each codon codes for one amino acid. tRNA brings in the right amino acid by matching codons with anticodons. Ribosomes move along the mRNA, read it with the help of tRNA, and help string amino acids to form peptides. At the end of translation, a peptide is formed and released. These peptides combine with other peptides to form proteins, the molecules that run life. And that's translation, turning genetic code into the stuff that makes us build muscle, digest milk, have a certain blood group, height, hair color, eye color. I can basically go on and on. Basically, proteins make everything that makes you, you.